and thank you everyone for joining us today. I'll go ahead and share my screen here, but what I'll say even before I start is that a lot of what you'll hear about tonight is relatively technical, and I hope you'll join us into a journey deep into the weeds of what we think about every day in my laboratory, and particularly the work that Michael, the graduate student who's going to talk about the specific work on this approach itself. I hope you'll join us for the very much the technical detail, because that's really what's required for some of the innovative approaches that we've brought to the table for tackling the pandemic. So even before I tell you, what the rest of the talk is going to be about, what I want to point out is that I'm a physician scientist by training, but really the work that I do in my lab is very much basic science. We're interested in fundamental problems of how life and biology work, and Michael is one of our graduate students that's interested in the same kinds of questions. And a lot of the technologies that we've been building over the years, both in my lab and across many other laboratories here at UCSF, are technologies that can be very rapidly used to find solutions to a whole host of problems, the SARS coronavirus being one of them. And what's exciting, the silver lining of this terrible period in history is that the scientific ability of almost all scientists has been unleashed to tackle this singular problem. So we'll give you our story on a unique perspective or a unique solution to the problem, and over the course of this series, you'll hear about a number of similar solutions for people coming at it from completely different lenses. So the premise of our project is to develop so-called inhalable personal protective equipment. The premise is that someone who's just recently learned that they're sick from the virus or is very early in the course of infection could inhale what's coming out of this little nebulizer that's shown on this title slide. And in this mist are molecules that are incredibly potent and effective at blocking the virus. So the story we'll tell you today is are why one would want to do this in the first place, and two, how do we even go about doing this? What are the principles that we use, and what does the journey look like? And who are the remarkable trainees that basically allowed this to happen? Just some quick disclosures. The one that's pertinent to the talk that you'll hear today is that both Michael and I are inventors on a patent filed by UCSF that pertains to this approach. And that's something that we have to do in order to actually get this to people eventually. All right, so how does the coronavirus actually work? And there's an enormous amount that's been learned about coronavirus biology over the past, certainly over the past 8 to 9 months, but really since the first coronavirus, since the first SARS virus, hit the scene almost 17 years ago. So I'm not going to get into all the nitty-gritty detail, but the one fundamental thing that I'll highlight is that for the virus to get inside your cells of your body, there has to be an interaction between a protein that's on the surface of the virus, and this protein is called the spike protein. The spike protein has to bind a receptor molecule on your cells, and that receptor we know is called, kind of a mouthful, it's called the angiotensin-converting enzyme 2 receptor, or ACE2 for short. But it's this one-on-one interaction between spike and ACE2 that's absolutely required for the virus to gain a toehold into your body in the first place. And once the virus binds this ACE2 receptor, it's taken up by cells, and that causes infection. So at this point, we know a lot about how this interaction happens. We've been able to map it to the most atomic level of detail, and we'll get into how we get the kinds of techniques that we use to really understand it at this level. But suffice it to say right now that we know quite a bit about how this spike protein, again shown in blue on the top here, binds this purple ACE2 receptor. So simply blocking this interaction between the spike protein and the ACE2 receptor is a very, very effective way of neutralizing the entry of the SARS coronavirus. And as a result, a number of the strategies that are being developed right now are either everything from vaccines to other therapeutics. Many of these strategies target this interaction and try to prevent it from happening. This gets into the notion of how your immune system does this normally. Now, and I'll walk you through a little bit of how the immune system recognizes viruses and the arms of the immune system that enable our bodies to fend off future infections. This is a beautiful slide rendered by Akiko Iwasaki, a real expert in virology at Yale University. And because the slide's so good, I just decided to use it directly, but credit to Dr. Iwasaki for this. So the idea is that when viruses infect cells of your body, they're recognized by a specialized set of cells called dendritic cells. And these dendritic cells, when they recognize viruses, turn on and move from peripherally in your body, let's say your lungs or in your nose, to specialized tissues called lymph nodes. And inside lymph nodes, these dendritic cells do this amazing thing where they tell two different types of cells called B cells and T cells to become active. And these two cell types are the weapons that the immune system has. So the B cells are really remarkable because their derivatives called plasma cells secrete molecules called antibodies. And these antibodies are very specific to a specific virus that has attacked you. So that's one way in which your immune system fights off viruses. A second way that your immune system fights off viruses is by so-called T cells. And these T cells are also very specific for specific viruses. And as a result, they can attack both the virus itself or virus-infected cells and control infection that way. So we'll really be focusing on the kinds of molecules that B cells make, these antibodies, to fight off infections. Now, antibodies are amazingly remarkable molecules. The cool thing about antibodies is that every time you see a different virus or every time you see a different pathogen, or your body develops a bespoke antibody for that pathogen. And the way that this happens is that each individual B cell has a different antibody that it makes. So for example, this B cell has seen some virus in the past and its antibodies are specific for that virus, whereas a different B cell makes antibodies that are very specific for a different virus. And this goes on and on. It's not just for viruses, it's for bacteria or for other pathogens as well. So how could this be? How can your immune system make these amazing antibodies that are super specific for basically anything that you might encounter, but on the flip side, have to basically deal with this enormous diversity? So it turns out the way that the immune system does this is by making antibodies that, by and large, are relatively similar in shape and structure, but part of their regions enable them to recognize almost an infinitude of antigens. So here I've just schematized this is kind of a cartoon depiction of what an antibody that's flowing in your blood or my blood looks like. And certain parts of these antibodies change their shapes to match the virus or the specific bacterial protein that they're trying to target. And in this schematic, each one of these differently colored blocks would be a different protein, from a different virus or a different pathogen. This figure also alludes to the fact that there's some shape complementarity. So for example, this antibody that's shown on the right here binds an antigen that has a specific shape but doesn't recognize an antigen with a different shape. And what's really amazing about the immune system is that even right now in your body or my body, there are at least 1 trillion different possibilities of configurational shapes of these antibodies. And that's what enables us to fend off basically any infection that we've seen in the past. And even what's more remarkable on top of this is that anytime you see a new pathogen, your body has the ability to make a completely new bespoke antibody against that pathogen. So that's really the premise of vaccination, is that eventually we want to be able to design a vaccine that mimics the proteins that are on the virus, such that your body makes your own antibodies to fight off infections. So that's one strategy to controlling the virus. Another strategy is, it often takes us quite a long time to make ideal vaccines that are safe. Another approach to doing this is, what if we could just identify really good antibodies, either from patients or from other sources, that are really good at turning off or inactivating the coronavirus? If we could do that, we could give those antibodies directly, and those would prevent infections or prevent people from getting sick. That general idea is called passive immunity. And the idea here is that instead of coaxing your body to develop your own antibodies, we just give you the antibodies directly. Now, there's two ways in which passive immunity is being applied for SARS-CoV-2 or for COVID-19 infections. The first idea is really quite old. The idea here, the premise is called convalescent plasma, but this idea goes back almost 100 years. So let's say there's some new pathogen that comes on the scene. Of course, right now that's coronavirus. And you have some patients that succumbed to the disease but got better. And that's basically shown here on the left. We know that these patients, as part of getting better, must have likely raised antibodies to control that infection. So what if we could simply take units of blood from those patients, people who've recovered from coronavirus and are willing to provide their own blood, and then harvest the part of blood that we know contains the antibodies, in this case plasma, and then give that to patients that are sick with the coronavirus. So again, this idea has been tried for other pathogens dating back to almost 100 years ago. And in general, it's a strategy that can really work in some instances. Now, the Food and Drug Administration provided an early use authorization for convalescent plasma in late August for hospitalized patients with COVID-19 infections. And this has been somewhat of a controversial decision, as one of the major scientific bodies, the National Institutes of Health, a panel led by that group, concluded that there's insufficient data to really recommend the use of convalescent plasma broadly. And while this is a great idea, the real challenge that is the safety profile of this approach is unclear, and part of the protocols for really getting this reliably to work remains unclear. So really what we need are prospective randomized trials. But the exciting thing long-term about convalescent plasma is that this strategy, if we can figure out the logistics, will always be available for any new pathogen. In contrast to convalescent plasma, an alternative approach to providing passive immunity are monoclonal antibodies, and this has been in the news a lot over the past couple of weeks. The idea here is that instead of taking the whole complex blood product of a patient who's recovered from coronavirus, what if we could identify one antibody that was just really, really good? And it was good from the perspective of neutralizing the virus, but it was also super stable. We could make it cheaply, all of these kinds of things. And we could identify that antibody, but instead of giving patients the blood product, we would make that antibody at remarkably large scale. We're talking, kilograms upon kilograms of that antibody, and then give that one specific antibody to patients infected with COVID-19. So this general strategy, a number of companies started working on almost at the start of the pandemic, and as a result, there are a number of monoclonal antibodies that are currently in late-stage clinical trials. Some of them have started to show some efficacy. One of these, for example, was given to Donald Trump when he was admitted for coronavirus infection. But really, the critical thing about monoclonal antibodies is that they have to be given through the blood, either for treatment or for prophylaxis. And one of the other challenges about monoclonal antibodies, specifically for coronavirus infections, is that because the infection initially takes hold in the nasal passageways and in the lungs, it's really important that the antibody have a high concentration or a high amount of protein in those spaces. But it turns out that antibodies delivered through the blood have relatively low exposure in those spaces. So only about 0.2% or 0.2% of blood-administered antibodies actually end up in the lungs. So what this leads to is really enormous doses of antibodies required to treat coronavirus infections. And as a result, what we're seeing now is that while these are really exciting potential therapies, the logistics of really being able to provide people these kinds of medications is unclear. On top of that, antibodies are relatively expensive to produce, and as a result, the broad utility of this approach remains a little bit unclear. So as part of our project, what we envisioned at the beginning when we started sometime in early March was an alternative approach to providing passive immunity. So let's say you had that same COVID-19 patient, but perhaps somewhat early in the course of infection. And instead of providing a neutralizing molecule like a monoclonal antibody through the blood, what if you could provide that same kind of approach, a molecule that would bind to the coronavirus proteins and completely prevent it from causing infections, but deliver it through either an aerosol that one could breathe in to really target the lung passageways, or via an aerosol or like a little nasal spray to target the nasal passageways. Now, the critical advantage of this approach is that it could be self-administered. Instead of patients having to come into the hospital or having to go to an infusion center, they could be given this kind of thing, and they could be at home or otherwise outside of the healthcare system and control their infections on their own. The other big advantage of this approach would be that because it's directly delivered to the site of infection, we could use far smaller doses than are being used for the monoclonal antibodies. Now, this doesn't mean that there's no challenges to this approach. One of the big challenges for delivering antibody-like molecules in this manner is that the protein itself would have to be incredibly stable. So it turns out that when you make these kinds of mists, the proteins themselves have to be so hardy that they can tolerate that, and many proteins just simply aren't stable enough to do that. On top of that, it's a little bit unclear how often one would have to dose this kind of these kinds of molecules through the airways, and a lot of those kinds of considerations are perhaps a little bit less worked out than the regular monoclonal antibody approach. Okay, so what do antibodies actually look like? I told you I'd get a little bit deep into the weeds on this. This was on the left. It was my cartoon that I showed you earlier on what an antibody looks like. What you're looking at in this very complex kind of diagram is actually the structure or the three-dimensional architecture of what an antibody truly looks like. Now, antibodies are complex proteins. They're made of two separate proteins that come together, a heavy and a light chain. And the business end of an antibody, again, this region that enables antibodies to bind essentially any kind of protein that you throw at them, is the so-called FAB region, and that's basically highlighted here. We can zoom in on that region and try to understand what enables antibodies, again, to recognize such a diverse array of potential pathogens. And antibodies, as a class of molecules, have what are called complementarity determining regions. So these loops that I've highlighted in different colors here are regions of the antibody that can vary enormously. And the variants or the completely different architectures in this region are what enable these antibodies to basically recognize all those different types of Legos. If we were to go back to this one slide, those loops, those complementarity determining regions, are these little cups right here. And the shape of these cups is then determined by the specific sequences and conformations of these CDR regions. So that's antibodies in your blood or my blood or any other human's blood. It turns out that camelids like alpaca, llamas, and other camels make these incredibly cool antibody-like molecules called nanobodies. And nanobodies are basically minimized antibodies. They're just this little piece of the FAB fragment. And instead of having 6 loops that allow them to recognize an infinite array of potential pathogens, they only have 3. But because they're far simpler, they have a number of advantages. One, they're very small. They're only about 1/10 the size of a regular antibody. They're incredibly stable, so they're very hardy to both heat stresses or completely drying out or even putting in an aerosol. They're very similar to human antibody heavy chains, so as a result, it's very unlikely that your own body will recognize these as something different. And because they're relatively simple, they can be rapidly produced on kilogram scale for relatively cheap. So with that kind of as a premise, we wanted to develop nanobodies that would bind to the spike protein of the coronavirus and prevent infections. And that these nanobodies we were hoping could be delivered to the aerosol, to the airways with these aerosols. Now, is there any precedence for this kind of approach? And it turns out that there is. There's a company called Ablink that had previously developed inhaled nanobodies for a completely different infection called respiratory syncytial virus. This is a virus that primarily causes infants to become quite sick and end up in the hospital. And what this company Ablink had done was developed a nanobody, so each one of these little blobs is a nanobody that's linked together to be 3 individual beads on a string that targets a protein very similar to the spike protein of coronavirus. And what they were able to show in test tube-based assays is that this molecule, which they called ALX0171, was very effective at preventing this virus from infecting cells. More importantly, this nanobody was stable to putting into an aerosol. And these are experiments that we do to understand whether once we aerosolize these nanobodies, whether they kind of clump together, and that's a really bad sign. And what you can see, these very sharp peaks are indicative that this specific nanobody doesn't do that. They went on to show in a preclinical efficacy model in lambs that this approach of delivering a nanobody directly to the airways was quite effective at preventing infections. And eventually in humans, they were able to show that delivery of this kind of approach was completely safe. And on top of that, had a half-life of about 20 hours. But what's most exciting is data that they just released for this approach about 2 or 3 weeks ago, which is they actually took this molecule all the way to testing in infants. And this is just data from their recently released study. So what you're looking at on the x-axis here is a time course after delivery of one of these nanobodies to these sick infants. And on the y-axis, what you're looking at is whether these investigators could measure culturable virus in the nasal turbinates or in the nasal passageways of these patients. So what's really exciting about this data is that the highest dose that they tested, which is in blue here, after a single dose of this approach, about 90% of patients had absolutely no detectable virus in their nasal passageways. Now, the sad thing about the study is that these patients, by the time they come to the hospital, are really quite sick. So although these molecules were very effective at preventing viral replication, they weren't as effective at preventing the clinical course of the disease. So the premise of the program that we're developing now is to really target very early stages of infection because we think that's where we will have the maximum benefit. Now, how are nanobodies normally discovered? Let's say you want an antibody against, SARS coronavirus or any other virus or any other pathogen. How do you go discover that? The classic way of doing this is highlighted on the top. What you do is you take a purified protein or some sort of inactivated virus, and you basically coax a llama or an alpaca to generate an immune response. You're basically vaccinating this animal, hoping that it will develop these cool antibodies, these minimized antibodies against your target. This process often takes about 2 to 6 months to raise that immune response. And then you can do a number of biotechnology approaches to go find those nanobodies within the animal that are specific to your pathogen, let's say in this case, the SARS coronavirus. My lab, over the past couple of years, has developed a completely alternative way of doing this. What we do is we make libraries of nanobodies. So what do I mean by that? What we do is we create sequences of nanobodies, in this case, billions of sequences of nanobodies, and then take baker's yeast and hook them up in a way such that each individual yeast has a unique nanobody on its surface. And here we've only depicted 5 of these. These libraries would have billions of individual nanobodies. And the idea here is that I told you earlier that your body has up to 1 trillion different antibodies that enable you to fight off pathogens. Our hope is that these libraries, these synthetic libraries of 1 billion variants, may have a couple that are specific to the SARS coronavirus or whatever other pathogen you're interested in. So the idea is then that we go fishing with this library. This is our pool, and we take a given protein that we want a nanobody against, and we go fishing in that pool of yeast and pick out the yeast, displaying a nanobody on the surface that we want. The key thing about this process is that it's incredibly fast. All of this library we basically have made in the past is readily available, and the process to actually find nanobodies against any specific target only takes us about 1 to 2 weeks. Now, the specific way that we did this for the SARS coronavirus protein is, again, we started with this pool of yeast, again, about 1 billion or so yeast, and we purified the spike protein from coronavirus, again, shown by this little blue gadget here, pulled on the spike protein to isolate those yeasts that are specific for spike, and amplified the yeast and repeated that process. Again, something that took us about 1 to 2 weeks to do. Okay, so the rest of the talk, again, is going to focus on how we developed nanobodies to block this interaction between spike and the ACE2 receptor. And that's something that Michael's really going to get into the weeds on. But before I transition to having Michael describe the research, I want to introduce 2 points that I think are going to be very critical for some of the detailed explanation that we'll get into. The first is, if we want to understand how 2 proteins bind, we need a framework for understanding that binding. So I can tell you that something is tight or binds very tightly to the spike protein or binds not so tightly. But how do we really quantitate that, and how do we get numbers on this? And what are the techniques that we use to really evaluate this? This is really important because in order for us to have a fighting chance, we need a protein that's going to bind. We need a nanobody that's going to bind incredibly tightly to a spike protein. So basically, once it binds, it never lets go. How do we know that that's the case? Here's a little bit of chemistry and chemistry recap for those of you who can harken back to those days. And I'm gonna give you the equation on the left, and then the sentence to describe what that means on the right. So if we want to understand 2 proteins binding to each other, in this case, let's call them a receptor and a ligand. This would be receptor molecule and a ligand molecule. They can come together to bind and make a receptor ligand. So that's just simple. R binds L to make RL. The way that we can understand this is we can describe once this reaction runs to equilibrium. So once it's gone forward, and then the amount that can go backwards, once that's all basically come to equilibrium, we can calculate a constant that describes how much of the RL there is and how much of R and L there is. And you can imagine something that binds very tightly. If R and L bind very, very tightly to each other, there'll be a lot more RL in there than separately R or L. That's just some relatively simple. But on top of that, we can actually also get at this a different way. If we, for example, had a way of asking how fast do R and L come together, and once they're together, how long do they stay together before they leave? If we had access to those 2 kinds of rates, we could also understand how tightly they bind. So for example, if R and L come together very fast, but once they're together, they take eons to dissociate or come apart, then we know that their affinity or their ability to bind to each other is very, very good. So again, these are words kind of describing on the right what I just told you. And the reason why this matters is we're going to show you a lot of data that directly tries to get at these rates. So how fast does a given nanobody bind to spike? And then once it's bound, how long does it stay bound to it? Now, the technique that we use to get at this is called surface plasmon resonance. That's, again, a mouthful. We'll probably just shorten it and call it SPR going forward. And the general idea behind this technique is that we can take a pretty complicated setup where there's a gold surface. But on that gold surface, we can put a protein of choice. So in all the experiments that we'll show you today, on the gold surface, there will be a spike protein. And then we can flow in a liquid solution some other protein, either a nanobody or some other protein, and ask how well does that nanobody bind to the spike that's immobilized on this surface. This will be represented as what's shown on the bottom right here, which is as binding happens, we get an increase in this resonance signal. And then if binding stops happening and decreases, then we know that things are falling off. So we can do this kind of experiment trying to validate whether purified spike protein directly binds the ACE2 receptor. This is just our first validation that we do in the lab. And so that's what this experiment looks like. What we're doing here in each one of these red curves. So again, the spike protein is immobilized on the surface of this chip. And as we flow more and more ACE2, again, that ACE2 receptor is what spike binds in our body, we can see that their signal builds up. And then at this dotted line, we stop adding ACE2, and that ACE2 now has a chance to fall off. So what we can then do is fit how fast this happens, how fast this protein comes on, and how fast it comes off. And those are the numbers you're going to see a number of times. And by using these 2 numbers, we can calculate a so-called KD, and that's the affinity. That's how tightly things bind. So the smaller this number, and this would be 44 nanomoles, this is 44 parts per billion molar, the tighter things bind. So this is a pretty good interaction, but Michael will show you numbers far, far, far better than this. Okay, so that's one kind of technique. That's how we understand if 2 things bind tight or weakly to each other. Now, the other kind of data that we're going to show you a lot of is we want to understand really not just whether things bind to each other, but how do they bind? What are the kinds of atomic interactions that happen that enable this binding to happen? And this is the kind of work that we do in my lab, and a kind of the nitty-gritty approaches that are required to really being able to do this effectively. And there's two techniques. If we want to understand life at the most basic level, we can't just put them under a simple light microscope. There are a number of techniques, and I'll highlight two of them, that allow us to really map where every single atom is in space. The classic way of doing this is a technique called X-ray crystallography. The idea here is if you can coax your protein of interest or your molecule of interest into a crystal, very similar to the crystals of sodium and chloride that you use for table salt. If you can coax them into a crystal, you can shoot those crystals with X-rays, and those X-rays interact with those crystals in an interesting manner to cause a pattern that you can detect with detectors. And using some well-worked-out math, we can use that pattern to then go find out where every single atom in this crystal is. So that's one way. That's the classic way of doing this. Over the past decade, there's been a revolution in a technique called cryoelectron microscopy. This is work in part done here at UCSF, where instead of having to coax these proteins into a crystal, we can directly freeze these proteins and image them instead of with an X-ray beam with an electron beam. And what we get at the end of the day are very, very noisy particles, very, very noisy images of the particles, of the protein particles themselves. But with new computational methods, we can average together these very noisy pictures to really get a three-dimensional view of what our proteins look like. So Michael will be showing you a lot of cryoelectron microscopy images that enable us to understand in atomic detail how our proteins actually work. Just to give you a little bit of a primer of what that data looks like, on the left here again is just one of those electron microscopy images. You can see this looks almost like static, but within this, what looks like static is actually real data. And this, for example, in this multicolored image is what the three-dimensional structure of the spike protein of coronavirus actually looks like. So again, this is like the 3D envelope of that structure, and you can see it's actually a pretty beautiful protein with this threefold symmetry. So with that, I'll stop, and what you're going to hear about next is our efforts to identify nanobodies that are incredibly potent for the coronavirus spike protein. Before I hand it off to Michael, I want to highlight that this was an amazing UCSF collaboration between my lab and Peter Walter's lab, work led by Michael, who you'll hear from, in addition to a number of really amazing and talented graduate students and postdoctoral fellows in my lab and in Peter's lab. And we're joined by an amazing group of collaborators, both here at UCSF, across the country in New York, as well as around the world, and Veronica especially is one of our amazing collaborators at the Institute Pasteur. A lot of the structural biology work that you'll hear about was led by a new way of doing science called the QBI Coronavirus Consortium, and you'll hear some of the amazing work that's come out of there too. So with that, I'll hand it over to Michael, and he'll walk you through what he's done and the team that he's been leading.
Thank you very much, Ashish, for that wonderful introduction. So, as Ashish mentioned, we did a screen using the Yeast surface display technology, and what I'll walk you through on the left side here is sort of in practice what we used, which is that we took purified spike protein, so the protein that coats the surface of viral particles. We incubated it with the yeast at 200 nanomolar concentration. We pulled out yeast that bind. We did it at a slightly lower concentration a second time in order to get higher affinity binders. And then we did it a third time at an even lower concentration to further enrich for high affinity binders. And then what we did was we took those yeast, and so you have a little test tube that's got your thousands to hundreds of thousands to billions of yeast, and what you can do is plate them all out, and then individual colonies will grow, and then you can pick those individual colonies, and what that represents is just like in the immune system where you have a B cell that has one antibody on its surface, this will be a yeast that has one nanobody on its surface. And from there, we can find the sequence of that nanobody, purify it, and test it to see, does it bind to the spike protein? What is the affinity that it binds to the spike protein? Does it block an interaction between that spike protein and ACE2? And so what we're seeing here in the middle graph is this SPR trace, the technology that Ashish just told you, where you immobilize spike protein on the surface and then you flow over a nanobody. In this case, it's a nanobody that I will refer to as Nanobody 6. And you flow it over the surface at varying concentrations, and you see that it goes on, binds to the spike protein very, very rapidly, and then it dissociates somewhat rapidly as well. And so when you look at this on rate and this off rate, you can calculate an affinity. And as we saw previously in the case of ACE2, which had about a 44 nanomolar affinity, this nanobody, called Nanobody 6, has about a 210 nanomolar affinity. So it's slightly worse, but in the same ballpark as the affinity that ACE2 has for spike. And one other interesting thing that you can do is you can purify specific domains of a protein. In this case, we purified the receptor binding domain, which is the portion of the spike protein that directly engages ACE2, the receptor on the surface of our cells. And what's interesting is, in this context, the nanobody, Nanobody 6, binds about 5 times more tightly than to the full-length spike, which speaks to the fact that this receptor binding domain can exist either in a hidden state or an open state. And this is biology that has been known previously, but it's interesting to see in our own hands that the accessibility of what you're binding to can impact the affinity that you observe. Moving on, what we then did was the second technique that Ashish mentioned, which is using cryoelectron microscopy in order to determine where exactly our nanobodies are binding. So what we saw previously was that we know nanobody, the Nanobody 6, is binding to the spike protein, and we know that it's binding to the receptor binding domain. But we don't know the exact orientation that it's binding. And so what we did was we mixed together your spike protein and your nanobody, take a bunch of these images, use computational modeling to figure out what this interaction actually looks like. And what we see is this nanobody, Nanobody 6, is actually quite interesting. And so it engages a primary RBD here, receptor binding domain, but it also makes some secondary contacts with a neighboring receptor binding domain. Now this is in direct contrast to another nanobody with similar affinity, but called Nanobody 11, which also binds to the receptor binding domain, but binds in a slightly different orientation. So as you can see here, it binds and sort of sticks upward and doesn't make any contacts with the neighboring receptor binding domain. So what we're showing here now is a zoomed in version of a top view and a side view of these two nanobodies. So Nanobody 6, which is engaging a primary RBD, as well as a secondary RBD, and Nanobody 11, which is engaging a primary RBD, but not engaging a secondary RBD. And what I hope you can see from this overlay that we have on the right-hand side is that both Nanobody 11 and Nanobody 6 are directly occluding binding of ACE2 to that receptor binding domain. So in the presence of Nanobody 11 and Nanobody 6, ACE2 simply can't interact with the spike protein, which is necessary for granting entry into cells. So what I showed you previously was the fact that Nanobody 6 binds to the spike protein, that it binds in a very unique way to the spike protein, and that it has a potency that is roughly on the order of ACE2 receptor. So how do we make this better? So one strategy that was employed by a similar nanobody technology by that company Ablink is you can trimerize it. And so what we did next was we took that structural data that we previously showed you, and we used this to inform how we were going to link up our discovered nanobodies. And so what we see when the spike protein is bound to nanobodies, when those receptor binding domains are all in the inactive down state, we see that the distance between ends of the nanobody is roughly 52 angstroms, which is on the order of about 15 amino acids, 15-ish amino acids. Whereas when a receptor binding domain is flipped into the open state, and this is a state that allows that receptor binding domain to directly engage with the ACE2 receptor, you see that it's larger. So if you can make a linker that is just the right size in order to engage when it's in the all down state, but not long enough to engage when it's in the all up state, not only can you block ACE2 from interacting with the spike protein, but you can also lock the spike protein into an inactive state. And there's further precedent for us being able to do this because of the fact that this nanobody, nanobody 6, engages 2 RBDs when it is in the all down state. This is in contrast to the other nanobody that we showed you, nanobody 11, which, whether it's in the down state or in the up state, either the closed or the open state, you see that the length between your 2 nanobody monomers, if they're engaging multiple receptor binding domains, is relatively similar. So what we would expect is, if you multimerize a nanobody that behaves like nanobody 6, we would expect it to drive major gains in potency. So what happens when you trimerize them or dimerize them? So what we are showing here are, again, those surface plasmon resonance traces, and you see that a monomer of nanobody 6 binds very rapidly and then dissociates very rapidly. However, if you make a dimer of nanobody 6, so a molecule that is capable of engaging 2 receptor binding domains, you see that it binds very rapidly. There's a slow phase that dissociates somewhat rapidly, and then a fast phase that dissociates very rapidly, and then a slow phase that dissociates very slowly. And then if you trimerize the molecule, you see that there are even more dramatic gains. So again, you have this fast phase with some rapid dissociation, but then there's a slow phase that's essentially a flat line. And what this flat line basically means is that a molecule, the trivalent nanobody 6, has engaged with the spike protein and is not coming off. It's stuck on there, and the affinity that we measure for this type of an interaction, what we were talking about previously was nanomolar. Below that is picomolar. This is probably into the femtomolar range. So this is dramatic affinity. This is stick and doesn't come off for over a week. So these are some pretty dramatic gains in potency. So one interesting thing was that fast phase that we saw here. What is that fast phase that we saw there? Is that some weird aberration, or is that something that's biologically interesting? And so, as I've mentioned before, a receptor binding domain can exist either in a down state or in an up state. And it interconverts between these two states, and when it's in the up state, it can bind to ACE2. Now, what we theorized was happening is that sometimes a receptor binding domain was in the up state. Trivalent nanobody 6 or bivalent nanobody 6 would engage with a receptor binding domain that is in the up state, and because of the distance between that up state and the down state, that trivalent molecule would not be able to fully engage with all the other receptor binding domains. So what we theorized was that this fast phase that we saw, where nanobody is rapidly falling off, what this represents is engagement of an RBD in the up state. So what we did was we again flowed nanobody over the surface of the spike, but we varied how long we flowed nanobody over the surface of the spike, anywhere from a few seconds to 10 minutes. And what we saw was that when you allow nanobody to continuously flow over the surface of spike, that over time, that fast phase of dissociation disappears. And what this means is that eventually that receptor binding domain that's in the up state will flip into the down state. And once it's flipped into the down state, then your trivalent nanobody, 3 nanobodies linked together, can engage all 3 receptor binding domains on a spike protein. And if you plot the percent slow phase versus fast phase as a relation to time, what you see is when you allow more and more and more time for the nanobody to associate, that fast phase basically disappears. And this is, I must say, this is a very cool experiment that we were very excited to see. That it's a really beautiful way to show kind of the mobility of a protein domain in a very unique way. And it speaks to the flexibility of the spike protein as well as the potency and mechanism of action for our trivalent nanobody. So basically what this is saying is, given sufficient time, a trivalent nanobody can bind to all 3 receptor binding domains, and it'll lock them all in the down state, this inactive state. So the big question now is, does this thing work? So what we've told you is, we found this nanobody, we showed you where it binds, and we told you that it binds pretty damn tightly to the spike protein. But does that do any good? And so we directly tested it in 2 different assays. So one assay, which is on the right, which I'll describe first conceptually, is, does it prevent live SARS-CoV-2 from infecting cells? The assay on the left is, does it prevent infection of this thing called pseudovirus? So what you can do in a pseudovirus assay is you take a different virus that isn't dangerous, but nonetheless has a different type of spike protein on its surface, and you replace that spike protein with the spike protein from SARS-CoV-2. So now what you've got is a not dangerous virus, but a virus nonetheless that has the SARS-CoV-2 spike protein on the surface of that viral particle. And you can use that to infect cells, and it can also be carrying a fluorescent marker that you can use to tell whether or not a cell has been infected or not. And so this thing is called pseudovirus, and the more conceptually straightforward thing is live virus, where you simply look, does this kill the cells? And so in the context of pseudovirus, I'll walk you through what all these traces are. This black line is ACE2 FC, which is basically a bivalent ACE2. So 2 copies of ACE2 fused together. And in gray, you've got monomeric ACE2. And so I hope it can be kind of conceptually straightforward as to why, if you mix in soluble ACE2, which we know bind to the spike protein, why that can sort of sequester these pseudoviral particles away and prevent them from interacting with the ACE2 receptor that is on your cells. And so your monomeric ACE2 will block your infection with about a 1 micromolar IC50. So if you have 1 micromolar of ACE2 in there, you'll block about 50% of infection. And then our monomeric nanobody 6, which we saw was a slightly weaker binder to ACE2, blocks in that roughly micromolar range, which makes sense. However, when you have a trivalent version of nanobody 6, this molecule that we showed you binds with exceptional potency. It has about a 1 nanomolar IC50. So you really need a very small amount of it. You need about 1,000-fold less of this trivalent nanobody 6 to block infection as you would with monomeric regular old ACE2, which is the thing that the spike protein was designed to interact with. And similarly, if you look at trivalent nanobody 11, which was that molecule I described, which doesn't quite have that optimal distance between the different monomers of the nanobody, which means that the linker isn't quite long enough to span it and allow full trivalent engagement, what you see is, again, that monomeric nanobody 11 is potent, but the trivalent nanobody 11 doesn't drive nearly the gain in potency that you see with this nanobody 6 molecule, which is optimized as a trivalent molecule based on the length of its linker. And if we now turn to a live virus assay, where basically what you do is you take your live SARS-CoV-2, and this was done with our collaborators in Paris, and it needs to be done under very special conditions, very high safety levels. What you do there is you take your live SARS-CoV-2, you mix it with your nanobody or not nanobody, and then you infect cells, and then you let it sit for a few days, and you see where are the live cells, where are the dead cells, where did viral replication happen, and more viruses popped out of that cell and then killed the cells surrounding it. So you can kind of look at a plate, stain it, and see where are there a bunch of live cells, where are there a bunch of dead cells. And again, what we see even more dramatically than we saw in the pseudovirus assay, which is always nice to see, is that trivalent nanobody 6 is a dramatic improvement and an incredibly potent inhibitor, about 100 picomolar in this assay compared to multiple micromolar for the monomeric version. So we're talking about a 5,000-fold improvement in potency and about a 200-fold improvement compared to a bivalent ACE2. So I hope everybody can appreciate how incredibly potent that molecule is, but we decided to make it even more potent. So one way that you can drive affinity is by multimerizing, as we previously showed. But another way that you can drive affinity is sort of through this classical affinity maturation technique where you optimize all the different amino acids on a molecule that are relevant for making contacts with the other protein that it's interacting with. And so what I'd like to describe now is that campaign, which is affinity maturation of nanobody 6. And what I'd like to show you here is comparing monomers and how we got them. So how do you get these? So what we previously did was we had a library with billions of unique nanobody sequences. And from there we pulled out a nanobody, which we termed nanobody 6, that was pretty potent, but we wanted to make it more potent. So what we did was we made a saturation mutagenesis library where we modified single amino acids in these loops that are important for recognizing the spike protein. We just change one to every single amino acid that it can be. Made a new library that has all these different variants in it, and then repeated the process and found what mutations made this nanobody bind even more potently to the spike protein. And what was very exciting is that 2 mutations floated to the top. So there was one mutation in CDR3, which engages the secondary receptor binding domain, and there was another mutation in CDR1, which engages the primary receptor binding domain. And in a little bit, I'll get into the weeds on kind of what those mutations actually did. But first, I'd like to show you the dramatic gains in potency that these mutations gave us. So, as we showed you previously, the monomeric version of nanobody 6 goes on really rapidly and comes off really rapidly, and it has about a 210 nanomolar affinity. However, when you use the affinity matured version of nanobody 6, what you see is it, again, goes on fairly rapidly, and it dissociates very, very slowly. And this is about a 500-fold gain in affinity that we generated by 2 simple point mutations in the CDR loops of this molecule, which is quite amazing, to be honest. You don't often see this. So the next thing that we did was we wanted to figure out how exactly are these modifications optimizing the affinity that this nanobody has for the spike protein. And so again, we turn to cryo-EM in order to determine a structure of matured nanobody 6, or MNB6, to the spike protein. And what you see in purple here is our matured nanobody 6, and as we can expect, it binds in a very similar manner to the parent molecule, nanobody 6. It binds to a primary RBD, and it has contacts with a secondary RBD. You do see that there are some minor changes that occur. The receptor binding domains are tweaked a little bit in their conformation, but what I'll show you next are sort of the major changes that arise when you modify nanobody 6 into the matured version of nanobody 6. Now in the upper right hand panel here, what we're seeing is the change in CDR1, where you have an isoleucine is modified to a tyrosine, and more or less the loop shape of nanobody 6 stays the same. However, this modification dramatically changes the local context. So it doesn't dramatically change, it optimizes the local contacts and is driving a gain in potency by kind of picking the best amino acid that you could have for that environment. Now CDR3, by contrast, is an entirely different story, where you have a proline to tyrosine modification that leads to a dramatic rearrangement of the loop. So this is about a 13 angstrom shift in position, and it's completely changing the contacts that this CDR is making, which is an entirely unexpected result that we were absolutely kind of shocked to see. But nonetheless, this is really amazing that it is driving these massive gains in potency through two somewhat unique mechanisms. One, an optimization of local contacts, and two, a complete rearrangement and changing of those contacts. And so what we ultimately made was a molecule that is very, very potent and very, very good at binding to SARS-CoV-2. But what we want isn't just something that binds to the SARS-CoV-2 spike protein. What we want is something that can be used as a therapeutic. And one thing that you must keep in mind when you're making a therapeutic is, just like if you have a virus that enters your cell and can activate the immune response, if you're going to add in a protein-based therapeutic, you want to make sure that you're not activating the immune response in the same exact way that a virus and viral proteins would be activating the immune system. So what we turn to next is a strategy called humanization, where we want to make this protein, this nanobody, look like something that the body wouldn't think twice about. So if you have a viral protein that comes into the cell, it's recognized as other. If we're going to put a nanobody into the cell, we want it to not think that it's an other. We want it to think that it's human and that it belongs. Luckily, nanobodies are already pretty similar to antibodies, to the heavy chain of human antibodies. And so what you can do in order to prevent an immune response, this immunogenic response, is you can transplant some of the amino acids that you would normally find, transplant the CDRs, these loops that recognize the spike protein, and put them onto the framework of a human heavy chain. So something that the body wouldn't think twice about seeing because it already sees it, but put these loops that we know interact with the spike protein, plop them on this framework that won't be immunogenic. The danger, of course, is if you're putting this on a new scaffold, will this in any way change the affinity that your molecule has for the spike protein? And we tested a few different frameworks, and we did see that some frameworks modified the affinity, but very excitingly, we found a framework that didn't. And so what I'm showing you here on the left is the affinity that the trivalent matured nanobody 6 has for the spike protein. So again, you flow it on, binds very rapidly. When you start your dissociation, it doesn't come off. So it binds so tightly that even if it's binding to an upstate receptor binding domain, that monomeric version can latch on long enough until it flips to the downstate, and then everything can engage in a full trivalent fashion. So this guy is binding, and it would take over 8 days, probably even longer, for one of these molecules to fall off the spike protein. For all intents and purposes, you can consider that a spike that is bound trivalent matured nanobody 6 will always have that molecule stuck to it. So critically, what we wanted to make sure is, does the humanized version of this molecule retain this exceptional potency, this amazing binding affinity? And so we made this humanized version that has an a framework that shouldn't be immunogenic. We put those CDRs that we know bind very tightly onto it. We purified that nanobody. We floated over the spike protein and monitored affinity by SBR. And excitingly, we saw that, nope, doesn't change the affinity. You can humanize this molecule so it won't be immunogenic, and it still binds exceptionally tightly to the spike protein. And so the next step is you've made this thing affinity matured. You've humanized it. You've trimerized it. Does this work? And so again, what we did was we infected cells with live SARS-CoV-2. And what I'm showing here is a comparison of the different generations of our molecule. And so in this light blue trace, what you see is original nanobody 6, which has about a 5 micromolar IC50. So it's going to take a lot of that molecule in order to neutralize SARS-CoV-2. If you mature the nanobody, so matured nanobody 6, which is this light green dashed line, so you've got a monomer that's now much, much more potent. So about a 500-fold, 200 to 500-fold increase in potency that you're getting from this affinity maturation campaign. If you trimerize these molecules, as we see in the solid blue and the solid green line, as we showed previously, you get dramatic gains in potency. And if you humanize it, which is what we see in this brown line, you really don't change your potency. And so there's two things that I'd really like to focus on here. One of which is that we're likely hitting the floor of our assay. So you'll run into a point where if you have, 1,000 molecules of SARS-CoV-2 that are infecting a cell, if you only have 100 molecules of your nanobody, you're not going to be able to block all of those SARS-CoV-2 viral particles from infecting cells, and that's going to be a scenario of ligand depletion. So we're in a regime of dramatic potency where really what's likely happening is you're running into a scenario where you're running out of molecules to do everything that you need. So I'm going to move on to the next part of our talk, which is basically just how potent our stuff is great, but how stable it is another thing that we really care about. And what I'm showing you here is basically on your y-axis is sort of a measure of your stability, and on the x-axis is the temperature. And what you see is that both for the monomeric and the trimeric version of our molecule, it takes very high temperatures, so 60 plus degrees Celsius, in order to cause your protein to unfold and sort of melt. And so that's one measure of stability. So another measure of stability is, can your molecule be aerosolized? And that's an important characteristic of aerosolized like this in a little mesh nebulizer. That's an important characteristic anytime that you want to deliver a molecule via nasal spray or via an aerosol. And so what we did in order to test this was we took our molecule, aerosolized it, collected that molecule, and then ran it over a column called a sizing column, which can basically separate things based on how big or how small they are. And critically, what we see is that either in the pre or post aerosolized form, you don't really see a dramatic change. So these molecules are a characteristic size, and when you aerosolize them, they stay as that size. They don't really aggregate up into the larger space and get all jumbled up and broken. Similarly, what you can do is lyophilize, which is a process where you kind of freeze dry. You basically suck all the water out and it turns into a powder. And this makes things really easy to ship all across the world because you can just ship it at room temperature. And you can similarly lyophilize it, so dry it down, take it wherever you want, resuspend it, same thing. Still very stable to that. And when you test it in virus neutralization assays, we also see that your molecule can be aerosolized, doesn't change your ability to neutralize virus. It can be lyophilized, and it can be heated. And so these are very important characteristics. You don't just want a molecule that's potent, you want something that's stable. And what I hope I've gone through is sort of the pathway in order to find a molecule that's really potent and also validate that it's really, really stable. And so I think I'll turn things back over to Ashish in order to sort of talk about the more clinical side of where we hope to go.
Great. Thanks, Michael, for going over that really remarkable set of data. I don't have a lot more to add. I think, I think Michael went through the deep nitty-gritty details of how we build a molecule like this. I think we're at a unique position now that we have a molecule that could actually start to get at this notion of providing passive immunity or providing an inhalable form of personal protective equipment. Hopefully what you're able to glean from Michael's discussion is that this molecule really is almost like a mousetrap. It binds to that spike protein with incredibly tightly, and once it binds, it never lets go. There's precedence for this kind of approach for other viruses for preventing viral replication. And so really what we think the unique advantage of an approach like this is for patients that are, again, very early in the course of infection, patients that are high risk, that are exposed to coronavirus patients like healthcare workers or nursing home residents, or other people that are generally high risk might benefit immensely from this approach to providing passive immunity. But with like many other projects in the academic setting, again, I want to remind you that this project wasn't really done by an industry group. This was done by a group of graduate students and postdoctoral fellows, Michael being a prime example of this group. The challenge for any of our academic groups to make this reality is crossing the so-called translational valley of death. So it's been known for some time that really great ideas that come out of the academic setting often have a hard time finding their way into practical utility. And this is an image that I like to use. This is a picture that my wife and I took coming back from Nevada, from the Grand Canyon into California. And this is Death Valley as you encounter it from the other side. And, really where we are in this process is somewhere, deep in the valley of death right now. We have some really great prospects for industry groups that are weighing whether it makes sense to enter into a new therapeutic for coronavirus. But long term, we think these approaches to tackling respiratory viruses, especially respiratory pandemics, is likely to have some value. And certainly for this pandemic, we think that this approach could really provide a very differentiated way of tackling the virus. So with that, again, I'll leave you with the acknowledgement slide so you can, stare at that. And with that, we'll, open it up to the question and answer session if people have thoughts or questions that we can tackle right now.
All right.
That's great. Thank you, Michael and Ashish. And Ashish, I don't know if you can see the Q&As.
Yes, I have them. Okay, great. So I'll just flip between myself and Michael for some of these questions. The first question is from Rosemary Juan. The question is, why were camelids chosen rather than a primate or a human? Thanks. Michael, you want to take that on?
Yeah. So in a camelid, you will have a very unique type of antibody. So camelids have a single chain antibody. So it just has a heavy chain that is responsible for engaging with an antigen, whereas humans will have both a heavy chain and a light chain. And so what a camelid antibody allows you to do is basically just clip off the top of that heavy chain, which we term a nanobody, and use that in order to recognize your antigen. And just as an aside, this was a very cool discovery that was not necessarily expected. So for the longest time, we thought that the heavy and light chains that humans and all other animals had was the only way to do it. And then it was discovered that camelids and also sharks have these single chain antibodies. So very unique thing that nature made, and we just take advantage of that.
Great, thanks, Michael. The second question is from Christian Chen, who asks, how long does it take to produce a usable dose of the humanized nanobody from the initial unmodified nanobody? I guess I'll take this. There's a process of engineering the molecule itself, and that process took Michael and the rest of our team about 3 months to do from start to finish, starting from one of our libraries. But even from having this blueprint of a molecule, and we can make laboratory quantities of this molecule, going from there to a usable dose for humans is a completely different ballgame. There's one thing, there's the kind of work that Michael or I or people in my lab can do at UCSF. But believe me, you don't want to be putting that kind of material into your body. There's a lot of very important regulatory work for any kind of protein therapeutic that has to be done in order to administer that to a person. So that's really the phase that we are right now, is to find groups that have that relevant expertise and the experience to take the really exciting molecule that we have in hand, but to really make it into a usable dose for testing in humans and eventually, hopefully, for actually therapeutic use more broadly. The next question is from Joanne Cagle. How do you define very early diagnosis, and at what point might this intervention not be viable? Michael, you want to take that on first, then I can add my own thoughts. But I think people will I think you'll find that answers to this question are probably pretty variable among scientists.
Yeah, this is a great question. I think it. I'll try to answer it as best I can, which is that as early as possible. So the moment that someone is PCR positive is when we would envision is the best case scenario that you would want to deliver this. So the moment you know that you have detectable virus in you, you'd want to start taking something like this, because every day that you allow the virus to replicate is going to be a day that this thing would get less and less effective. And I'll take it to an extreme and say, once you have a full-blown infection and your lungs are glassy and you've got all of these issues, something like this, which is designed to prevent replication of the virus and viral entry, isn't going to be very effective. So another group of individuals that this could be very effective in is people who it's even before they've been diagnosed. So let's say your roommate or someone you live with or interacted with was diagnosed as being infected. Then you could potentially start taking this molecule as a prophylactic in order to nip it in the bud. Ashish, anything to add on that?
No, that's great. I think we have evidence now from the respiratory syncytial virus data that that other study that I mentioned from Ablink said really targeting early infections is important. And this is almost certainly going to be the case for some of the monoclonal antibody approaches as well. We think that the unique ability of enabling patients to self-administer this to themselves at home may enable really targeting very early stages of infection. If you think about it right now, what happens to a patient that just learns that they've either been exposed to someone or are in the early stages of infection? We largely tell them to quarantine until they have symptoms that are severe enough to warrant coming to the hospital. So we think that, again, this approach, because it's so patient-friendly, may enable us to help patients who are pretty early in the course of infection. And that's where all of these viral entry inhibitors will have their most will be most effective. Great. Next question is from Spencer Robinson. An amazing amount of work produced under extraordinary circumstances. The term, the comment is really on the term inhalable PPE. And really, when we think of PPE, we think of materials that prevent the entry of the virus into our system. And so these molecules aren't really inactivating these molecules are inactivating the virus, not really preventing their entry. Any comments in the name of inhalable PPE? So I'll take this, and Michael, you can add if you want. We've tried to, one of the big challenges in scientific communication is finding ways of enabling people to engage with what we've actually done. So we agree that the perspective that you're providing here, that it's not really a form of personal protective equipment, but in some ways the way we view it, in the kind of the very ideal scenario, let's say an approach like this is very safe and broadly deployable, it might provide an adjunct to PPE that's even far more effective than PPE. This isn't to say that it would replace PPE, but it would be, let's say you are going to get on a long-haul flight from here to Australia, and you want some way of decreasing your odds of getting sick. In addition to the mask that you'd wear, perhaps a dose of an AeroNab would really provide that kind of protection. So really the premise of using the inhalable PPE term was really to engage with people and with the experience that they currently have. But really, we think of this, as a therapeutic, and in the ideal setting as a way of really decreasing the probability of getting sick. Michael, you want to add any other comments to that?
I think that, handles it. It's just a way to engage people and get them describe a fairly complicated thing in something that they've heard already.
Great. Next question is from Corey Silver. The question is, how many other labs are working on similar efforts, and is there an entity, e.g. like the CDC, that is enabling collaboration across the field? Is the competitive process working here, or would a stronger arm from the government be preferred? So there are a few other laboratories that are working on trying to find academic groups and industry groups that are trying to find inhaled therapeutics and in specific inhaled antibody-like molecules. We've benefited enormously in our own work from collaborating with some of those groups. And there's a healthy dose of competition. We think our molecules are great, and there are great other molecules that are progressing as well. The CDC has not been really the group that's been driving any kind of collaboration, mostly because they focus on public health. By contrast, the National Institutes of Health has really been quarterbacking a lot of the efforts to coordinate various groups that want to run clinical trials in the COVID-19 space. And we've had great conversations with some of those groups to plot what the ideal path forward may be.
Ashish, we've got one other one. We've got anonymous attendee that says, given this may end up in users' hands, is there any risk of overdosing on these nanobodies?
Michael, you want to take that?
Yeah. So broadly speaking, no, I don't think so. What we've seen from a lot of our data is that you can give really large amounts of these to cells, and it doesn't really seem to impact them in the same way that, say, if you give a large molar quantity of remdesivir to a cell, that's in the context of cell culture. If you want to actually see what is a safe dose for a person, this will need to be borne out in human trials, but based on past experience with antibody dosing and with other nanobody therapeutics, there doesn't really seem to be any worry about overdosing in that sense.
The only thing that I'll add to this is that at a very simplistic level, this molecule is designed to be incredibly specific for the virus. Unlike a lot of other therapies that people are taking forward that are trying to change something about the human body, either the immune system or some other aspect of human biology, this these nanobodies are meant to be very, very specific for the virus. So as a result, we think that the probability of having some sort of safety problem is relatively low. But again, we don't, that's why we do clinical trials. We have to have real human data before we could ever think about, providing this more broadly. There's a question from Clara Hancock in the chat, which I'll take. What are the next steps for this approach? What is the timeline for bringing this therapeutic to market? And I think along those same lines, Mary Murphy in the chat asks, how long did it take the RSV company to bring their drug to use in humans? What would be the best case scenario for your product? So this is, I think, I'll group all of these kinds of questions, which is how fast can you get there kind of questions. So the real limitation to bringing this to market is, from our perspective, finding an industry partner who has the ability and more importantly, the resources, both the financial resources and the personnel resources with the right kinds of expertise to do what's practically required for drug development. So that's everything from how do you scale something like this, safely to make kilogram quantities of it? How do you go through the kinds of preliminary safety studies that are required by the FDA to even start a clinical trial? And then once you want to test a molecule like this in humans, what is the ideal clinical trial to do? It's going to be very hard to run a trial that's incredibly large that enrolls thousands of people like we have to do with the vaccines. By contrast, you want a large enough trial with the right kinds of patients to make sure that if it's going to be effective, that you're going to have a signal there. So again, these are all things that are outside of the realm of our direct expertise, and that's the kind of partner that we're deep, in discussions with right now.
We've got two more in the chat that I'm going to group together, which is, were there any notable benefits of Nanobody 11 as opposed to Nanobody 6? And would virus mutations affect the efficacy of the pathway you've described? I'm going to group these two together in order to say that, were there any notable... I'll answer the second one first, which is that coronaviruses thankfully mutate much more slowly than a lot of other viruses. Are there mutations that might become an issue? In terms of mutations that are rapidly taking over the globe, one that you might have heard of is this D614G mutation. That wouldn't in any way impact the ability of this nanobody to bind. There don't seem to be any receptor binding domain mutations that are rapidly taking over the population. That being said, escape variants are something that you always need to be considering in viruses, which is if you give something that is meant to kill the virus or stop it from replicating, is there a way that that virus can escape that? You always need to be keeping that in mind. And so in order to get at that question, which is what are the benefits of Nanobody 11 or another nanobody, we have some other nanobodies that we didn't discuss that also target the virus. In terms of potency, Nanobody 6 was the best from a potency standpoint, but the benefit of other nanobodies is that you can make a cocktail in order to prevent the issue of escape variants. That being said, because coronavirus mutates so slowly and it doesn't seem like that there are any receptor binding domain mutations taking over the population, we're not terribly worried about this, and the ability to generate a new version of Nanobody 6 pretty rapidly also gives us comfort that if something were to arise, I think that we can address it on a reasonable timescale.
Great. So I think that tackles the questions that at least I have visible.
Yeah, I think that was fantastic. Thank you guys so much. Let's just hope that either the government gives you $1 billion to bring this to market, or there's a company you find that wants to invest in the process. So again, thanks very much. That was great, and looking forward to more great work coming from the labs that you're running. So, all right, gentlemen, take care. You've been a great audience. Thank you so much. We'll see you in the coming weeks. We'll get to know each other well, and as we continue to learn about COVID-19. Take care now.
Great. Thanks, everybody. Thanks
Thank you
for having us.